Our group topic is Shigella Sone. We have 8 group members. They are 徐宇航、陈景哲、刘维志、蒋静文、梁烨、Taswani、尼尔贾、Akila. This is our contents.
Hi, my name is Nur Akila Batrisha. My matrix number is A209970. The pathogenic microbe that we choose is Shigella sonii. So now I would like to talk about the introduction of the pathogen. Shigella sonii is a species of Shigella. Together with Shigella flexneri, it is responsible for 19% of Shigellosis cases. Shigella sonii is named for the Danish bacteriologist Carl Olaf Sonne. Shigella are bacteria that cause diarrhea. The most common ways people get sick are from eating or drinking contaminated food or water and contact with someone who is sick or has recently been sick with Shigella sonii. It is a gram-negative, rod-shaped and non-motile. This bacterium does not retain the crystal violet stain and lacks of flagella for movement. Other than that, it is facultative and it can grow with or without oxygen. It does not form resistant spores, meaning it is heat resistive and generally killed by proper cooking and pasteurization. The host specificity are humans and higher primates. These are the only known natural reservoirs of Shigella species. The biochemical properties for Shigella sonii is non-lactose fermenters and does not decarboxylate lysine, which helps distinguish it from other bacteria like E. coli in lab settings. In food safety, Shigella sonii have low infectious dose. The extremely small number of bacteria required to cause illness means that even minor contamination can lead to an outbreak. This making good hygiene is the most important thing. Next, transmission via food handling. The primary route of food contamination is the fecal-oral route, often through infected food handlers with poor personal hygiene who touch food or preparation surfaces with contaminated hands. It is also survival in food. Shigella sonii are acid resistant and salt tolerant. It can survive for extended periods in various food including low pH foods like fruits and vegetables, prepared salad, and refrigerated items. Lastly, it is outbreak potential. Due to high communicability and low infectious dose, it is a major cause of sporadic and common source of outbreaks in developed countries, particularly in institutional settings like daycares and restaurants.
Shigella sonnei produces several types of toxins and pathogenic metabolites that contribute to its ability to cause shigellosis by damaging the intestinal tissues, disrupting normal gut function, and provoke the strong inflammatory responses. These can be classified into cytotoxins, endotoxins, enterotoxins, and pathogenic metabolites. Cytotoxins are toxins that directly damage or kill the host cells. In Shigella sonnei, Shiga toxin is not commonly produced, but in rare cases, some strains may acquire Shiga toxin genes through the bacteriophages. When present, this toxin inhibits the protein synthesis in intestinal epithelial cells by inactivating the 60S ribosomal subunit that leads to the cell death, ulceration of the intestinal lining, and bloody diarrhea. This cytotoxic activity contributes to severe mucosal damage and enhances the inflammatory process in the colon. Next is endotoxins, which are mainly lipopolysaccharide, are a constant and important virulence factor of Shigella sonnei. Lipopolysaccharide is a structural component of the outer membrane of the bacterium and is released when the bacterial cells die or multiply. It strongly stimulates the host immune systems, triggering the release of inflammatory cytokines such as the interleukins and tumor necrosis factor. This inflammatory response is responsible for fever, abdominal cramps, and inflammation of the intestinal mucosa, and it plays a major role in the development of diarrhea and systematic symptoms of shigellosis.
The third one is enterotoxins affect the intestinal epithelium and alter fluids and electrolytes balance. Signal enterotoxin 1 increases intestinal secretion of fluids. Signal internal toxin 2 acts synergistically with bacterial invasion, contributes to watery diarrhea, especially during the early stages of infection before extensive tissue damage. The last one is pathogenic metabolites, enhance survival and virulence within the host. Cytophils that allow the bacterium to obtain iron. Acid resistance metabolites that help the organism survive passage through the acidic stomach. Enzymes that protect against oxidative stresses generated by host immune cells do not directly cause tissue damage. Essential essential for bacterial multiplication, persistence, and effective expression of toxin-mediated pathogenicity.
I will briefly apply how the inherent properties of food itself influence the risk from Shigella sonnei, a dangerous pathogen causing survivable illness. The key point to remember is that this bacteria doesn't need to grow into high numbers, just a tiny amount, less than 100 cells, can make you sick. The first one is pH. Bacteria generally prefer neutral conditions, but Shigella sonnei has a critical survival skill, acid tolerance. This lets it survive stomach acid to infect the gut. In food, this means it can persist and even slowly grow in many common items with a pH above 4.5, like cut melons, milk, or cooked rice, while very acidic foods like vinegar or yogurt can suppress it overcome. A brief contact won't necessarily kill it. The second one is water activity. This measures how much free water is available for microbes to use. Shigella sonnei needs a lot of it. It thrives in high moisture foods like salads or wheat ingredients. In very dry food like yogurt, it can't grow. Here's an important nuance. In most foods, think species or powdered milk, the bacteria can enter a dormant state. It's not growing, but it's not dead either. Once consumed, it can working in your intestines and cause infection. So even raw foods can be risky if contaminated. The third one is nutrients and food structure. This bacteria isn't picky. It can use simple nutrients to grow rapidly. Protein and carb-rich foods like potato salads, dairy, or poultry are perfect breeding grounds. Also, its physical form matters. Shigella can live with or without oxygen. More critically, process like chopping or mixing can take a small surface contamination and spread it throughout the entire dish, turning a local problem into a widespread one. The first external factor is temperature. Temperature is the primary factor determining the fate of Shigella. In the danger zone between 20 and 40 degrees Celsius, such as in food stored at room temperatures. The bacteria can survive and grow rapidly. High temperatures above 60 degrees Celsius cause rapid inactivation, making raw heating an effective control measure. Freezing, while not immediately lethal, significantly inhibits bacterial activity. The second external factor is humidity and atmosphere. Humidity is another critical factor. Shell gather through in most environments like damp surfaces, fresh produce, and wet packaging. However, low humidity and dry conditions lead to rapid dehydration and cell death, while shell gather is facultative and can survive with or without oxygen. Modified atmospheres with low oxygen and high carbon dioxide can effectively suppress its survival. Third, physical and chemical stress. Shigella is highly sensitive to environmental and chemical stressors. It may persist in untreated water or environments with minimal microbial contamination. However, common measures like UV light, disinfectants, aqueduct conditions, and even mechanical washing can effectively disrupt its integrity and reduce viability.
One, core transmission route, fecal-oral transmission, bacteria in the feces of infected individuals, contaminate media such as water sources, food, tableware, and hands, then enter the healthy human body through the oral cavity. This is the main logic behind food contamination by the passenger. 2. Specific scenarios of food contamination. Water source contamination. Drinking water contaminated with feces is directly used for food processing and ingredient cleaning. Processing stage. Food handlers carry the pathogen and contaminated food due to poor hand hygiene. Ingredient contamination. Contaminated manures is used in planting or breeding, leading to pathogen adhesion on fruits, vegetables, aquatic products. Cross-contamination. Raw and cooked foods are stored together, and kitchen utensils are not served.
Passenger mechanism. The first one is adhesion and invasion. 1. Intentional adhesion. Shigella something adheres to the surface of intentional cells of the human chronic mucosa via structures such as fibria and brains praxillus to specific receptors on the intestinal epithelium. 2. Cell invasion. After being intercepted by epithelium cells, the bacteria proliferate intracellularly and spread to adjacent cells, damaging the integrity of the intestinal mucosa and integrating mucosal inflammation and injury. The second one, toxin release and pathological reactions. 1. Shigella toxin. Some stress of Shigella zone secretes Shigella toxin, which inhibits protein in synthesis in host cells, leading to necrosis and shedding of intestinal mucosal cells and ulcer formation. 2. endotoxin effect. Endotoxin relaxed upon bacterial death and lashes stimulate the intestinal mucosa to induce an inflammatory response. It can also enter the bloodstream and cause systemic symptoms such as fever and toxic shock.
Let's talk about disease types and symptoms. For the clinical symptoms, the including period is usually 1 to 3 days and short and a few hours and up to 7 days in some cases. And for the typical symptoms, first is intestinal symptoms, abdominal pain, diarrhea, and prominent tenderness. Systemic symptoms, fever, fatigue, loss of appetite. Severe case may experience dehydration and electrolyte imbalance. Severe manifestations. Children or immunocompromised individual may develop toxic dysentery, presenting with hyperpyretic convulsions, disturbance of consciousness, septic shock, which can be threatening.
Infants, young children, and immunocompromised individuals, including the elderly, are at highest risk. Their symptoms are often more severe. Those in areas with poor sanitation or collective settings like school also face significantly greater risks of infection and outbreaks. Thank you, everyone.